Okay, hi everyone. Welcome to this new ESTR e-seminar. My name is Sandrine Dubrac, and with my co-chair, Kilian Eyerich, we will moderate this new ESTR Kitchen Freshly Baked session, supported by BMS and sponsored by Almiral. Today there will be two presenters, Elodie Labit and then Longlong Luo. We will have a 10-minute presentation each, followed by a Q&A after the second talks. And there is also a chat option for those who wish to use it. So the first paper is presented by Elodie Labit. Elodie did her PhD in the lab of Luis Castella in Toulouse, and she obtained it in 2016. Then where she studied the negative impact of opioid during inguinal adipose tissue regeneration. Then she spent a year as volunteer scientific in Madagascar before joining the lab of Jess Birnaski in Canada at the University of Calgary, where she followed up on her work on tissue regeneration and wound healing. And this is actually the topic of the paper she will present today, published in Cell last December. And recently, Elodie went back to France in Toulouse to join the lab of Nicolas Gaudencio, where she works on skin wound healing. The title of her paper that she will present now is Fibroblast Inflammatory Priming Determines Regenerative versus Fibrotic Skin Repair in Reindeer. And Elodie, the stage is yours to present you this very exciting paper.
Okay. I hope you can see my screen now.
Yes, we do.
Okay, cool. So first, thank you, Sandrine, for this kind introduction. And first, I would like to thank the ESDR organizer for giving me this great opportunity to share this data to this fresh baked session. So today, I will talk about wound healing. But first, I will talk about homeostasis. So what is tissue homeostasis? It's a fine equilibrium responsible for the maintenance and function of each tissue, and this is due to the tight communication between stem cell, epithelial cells, stromal cell, immune cells. And we know that after an injury, this fine equilibrium is disrupted. We also know that only the lower vertebrates, like salamander or axolotl, have the abilities to regenerate and refine this fine equilibrium. Instead of regeneration, adult mammals develop other kind of repair, and we develop scar or fibrosis or chronic wound. It's important to note that there is an exception, the fetus, the mammal fetus, that is able to regenerate as well. So skin, like other tissue or organ, are not able to fully regenerate in adult mammals. And we know that fibroblasts play an important role in wound healing and scarring and fibrosis. And we know that there is a different kind of fibroblast with different origin, different lineage. And we and other labs have found that wound responsive fibroblasts activate different transcriptional program and chromatin configuration in order to enable regeneration. And this work have predominantly what was done in a rodent model. But one question remains: what drives dermal fibroblasts to become scar-forming rather than regenerative? And for that, and for this story, we use the reindeer. Why do we use reindeer? So first, because each year they are able to generate new antler. So the antler is this structure that you can see in the head. We also know that if we ampute some branches, the antler can regrow or regenerate. It's very important for us because antlers are covered by skin that we will name velvet, and also because the reindeer is more similar in terms of wound healing of human in comparison with rodent models. So before starting to dig into this cell paper data, I would like to say that these data that I will share today are mostly due to the very hard work, more than 1 decade of work from 2 PhD students, so Oli and Sartek, and under the supervision of our amazing supervisor or PI, Jeff Biernaski. So what we exactly did? So we did wound into the back skin of the reindeer, and we compare the wound healing of this wound with wound that we did in the velvet that covers the antler of the reindeer. And you can see here, so this is the kind of wound that we did with a punch biopsy on the back skin. And as I told you earlier, 60, 30, and 60 days after the wound, we have a scar or a fibrosis in the back skin. However, when we are doing the same kind of biopsy or injury in the velvet, you can barely see the wound at 60 days after wounding. And when we compare the histological picture, you can clearly see the difference between the velvet in the bottom and the back skin. And here you have the quantification. So we can say that the velvet is regenerating because there is no gland, there is hair follicle, and there is also proliferation that you can see here with the staining of the Ki67. So the next question that we tried to reply was this one: does the antler provide a permissive environment to enable velvet regeneration? And for that, we did this experiment. So we took some velvet from the antler that we grafted the back skin of the reindeer. We waited 30 days, then we did a wound of this velvet grafted into the back skin. Then we study this grafted part 30 days after the wounding. So here it's before wounding. You can clearly see the difference between the back skin and the ectopic velvet graft. And when we are looking 30 days after wounding, this grafted velvet. So here you have the margin of the wound stained with the fluoro bead, and you cannot see any difference between the ectopic graft and the wounded area of this ectopic velvet graft. And here you have the quantification. So we were interested by understanding what is the main difference between the velvet and the back skin in terms of cell type, for example. So that is why we did single cell RNA-seq at D0 in the back versus in the velvet skin. And you can clearly see here in this UMAP plot that we have exactly the same cell type present in back and in velvet. But when we did the Pearson correlation to see which cells are most discrepant between the velvet and the back, the most different cells are the fibroblasts. So that is why we studied the fibroblast. And if you remember what I told you in the introduction, I told you that the fetal mammals were able to regenerate, whereas the adult mammals are more prone to fibrosis. So we compare the fibroblast, the dermal fibroblast, from human, fetal human, and the velvet, so the skin that is able to regenerate in the reindeer. And we also compare the adult dermal fibroblast with the back skin fibroblast. And what we have found is that fetus or fetal dermal fibroblast and velvet fibroblast shared a lot of conserved regenerative program. So they express, for example, CRAB-BP1 or periostin. The adult dermal fibroblast and the back skin fibroblast from reindeer share more pro-inflammatory, pro-fibrotic program, and is the case, for example, for IL-6 or the prostaglandin gene. So what we did here, we took all the fibroblasts at different time points after wounding, so D0, D3, D7, and D14 after wounding in back and in the velvet skin. So this is the UMAP projection. And as I told you, we see a difference in fibroblasts at D0 in back and in velvet. So you see they don't have the same localization in this UMAP. It means they are very different. And it's very interesting because 7 days after the wound, the thing that we can see here is that the fibroblasts from velvet and the fibroblasts from back are looking pretty the same. But at 14 days, they are looking very different. In fact, at D14, the back skin fibroblasts stay exactly like at D7, but the fibroblasts that are responsible for regeneration, so from velvet, seems to come back at their place that they have at D0. Furthermore, you can see that the fibroblasts from the back express ACTA2 or alpha SMA for the protein. So this is a marker for fibrosis or myofibroblast. And the fibroblasts from the velvet, so the regenerative fibroblasts, express CRAB-BP1. So this is a marker for embryonic fibroblast development, for example. The next question that we try to reply is that is there a comparable pool of circulating immune cells that are supplying the velvet and the back skin? For that, what we did, we took, so we did the wound, and 3 days after the wound in back or in the velvet skin, we took the blood that we find close to the velvet, so in the velvet vein or in the back vein, and we did single-cell RNA-seq. You can see here that it's not very surprising because it's the same animal, so it's normal that we have the same blood everywhere, but we did the experiment, and we do not see any difference in terms of immune cell repartition in these two veins. And here you have the maturity of the different immune cells that are in the blood, in velvet or in back skin, and we do not see any difference in terms of maturity of immune cell. So here what you have is that it's a UMAP where we have all the immune cell and fibroblasts and keratinocytes from D0 until D14 after wounding. And when we are looking only the PTPRC positive cells, so the CD45 positive cells, the immune cell, we can clearly see that in the back skin, so in blue, we have an amplified and a prolonged immune response only in back skin in comparison with the velvet skin. So it seems that the fibroblasts are able to change the immune milieu and the immune response after wounding in reindeer. The next question, was, are the fibroblast fates at ground states are determinating for the milieu after the wounding? So for that, we took back or velvet dermal fibroblast, and we added in co-culture some peripheral leukocytes that we took in the vein 3 days after wounding. So this is a single cell that we did in blood skin, so without co-culture, and we see that they are looking exactly the same. However, when we co-cultivate the blood with the back fibroblasts, and when we compare them with the blood culture with the velvet fibroblast, you can clearly see that the blood are totally, the cell blood are totally different. Finally, we try to understand how the fibroblasts are communicating with immune cell to modulate the wound repair response. So we did cell-cell connectomic, and what we found, so here you have the fibroblast, here you have all the immune cells, and here are the ligands that could be sent by the fibroblast, and here it's the receptor present in different immune cell type. So this it's in the velvet, and this here it's in the back. And what we found is that the fibroblast from the fibrotic place, so the skin, are able to secrete or should be able to secrete CSF1, for example, or 6CL12, so pro-inflammatory cytokines, whereas the regenerative fibroblasts, so in the velvet, are more prone to produce midkine or tenacin, for example. And finally, what we tried is to block this signaling, pro-inflammatory signaling pathway, and to see if we can convert a fibrosis into a regeneration.
Elodie, you have to finish the talk because the 10 minutes are over. Yeah, okay. Thank you.
So, and what we found is that when we block the CSF1 or the 6CL12, we have more regeneration, and this was done in rodent mice. So I think you all understand the model, so I will finish here by thanking all the Birnaski lab members and for your attention.
Thank you very much, Elodie. That was really impressive. And as we said, we discussed this all together with the second presentation, which will follow directly now. So I would encourage all of the audience to please write any questions in the chat. We will stay in the topic of wound healing, and now Dr. Longlong Luo from the Uppsala University will present his data on translational candidate factor in wound healing. Dr. Luo is a member of the famous working group of Enniko Sonkoli, previously at Karolinska and now at Uppsala, who's working in this field of non-coding RNAs, in particular in the context of inflammatory skin diseases. So that's your deviation from wound healing to metabolic activity and psoriasis and inflammation now since years. And we are very much looking forward to your presentation now.
Thank you for your kind introduction, and hopefully everyone can hear my voice and see the screen. And thanks ESDR for offering me this opportunity to present my work here. Today, I'm going to talk about non-coding RNA and its rules in psoriasis. As a chronic inflammatory skin disease, psoriasis is characterized by shapely erythematosis and scaly skin and thickened patches. The IL-23 and IL-17A pathway had been investigated and identified at the central axis in the pathogenesis of psoriasis, which is mediated by the disturbed immune cells and keratinocytes. Activated keratinocytes secrete a series of the inflammatory mediators to sustain the inflammation response and lead to the epidermis hyperproliferation, indicating that the keratinocytes are not only the positive bystander, but also play important and active roles in the psoriasis disease. To begin, answer the question what non-coding RNA are, I will go back to the basic and present the flow of the genetic information in the cells, which start from the DNA that transcribe to RNA and then translate to protein. Non-coding RNAs, or link RNAs, are formed during the processing of the RNA transcription. And most of the link RNA are transcribed by the polymerase II. They are structurally and functionally diverse transcripts with over 200 nucleotides in length, with no or limited capacity to encode the function peptides. There are over 25,000 of the link RNA in the human genome, and their transcripts can be found in the both nuclear or cytoplasma or separately. Noncoding RNA has been shown to regulate the gene expression by a variety of the mechanisms, including the gene regulations of the mRNA splicing stability and the translation, moderation of the epigenetic status by recruiting the chromatin modification proteins, recruit or blockade of the transcription factors, or decline by others RNAs or proteins. However, little is known about their roles in the epidermis function and the skin disease. Dysregulation of link RNA has been shown before in full-depth skin from the psoriasis. This reflects the expression derived from the mixture of the different epidermis and dermis cell types. Here, we would like to review cell-specific changes in the expression, especially for the keratinocytes. In this study, we applied 4 milliliter punctus biopsies, which collect from the healthy donors and from the lesional and non-lesional skin from the psoriasis patient. Then we separate epidermis from dermis and then triplet the cells into single-cell suspension. Epidermis CD45 negative cells are merely keratinocytes. They are sorted while the MAC sorting. In the end, we analyzed the transcription profiling of the link RNA in these keratinocytes. Link 00958 was one of the most upregulated link RNA from the psoriasis lesion. This function in the skin has been unknown before. We perform RT-qPCR and a single molecular in situ hybridization and confirm its significant increased expression in the psoriasis lesional epidermis. The subcellular localization of the link RNA can provide the clue of their function. We performed confocal microscopy in the keratinocytes and found there's a relative more enrichment of the link 00958 moleculars in the cytosolic compartment. In line with this, we also perform RT-qPCR analysis separately in nuclear and cytosolic fractionation and shows that link 00958 is mainly localized in the cytoplasm. Next, we aim to identify which kind of factors responsible for the overexpression of the link 00958 in psoriasis keratinocytes. We use the series of psoriasis-associated inflammatory cytokine like IL-1 beta, IL-22, TNF-alpha, and IL-17A to treat keratinocytes, and only found IL-17A significantly induced these transcripts already after 3 hours. We also analyzed the effects of the IL-17A on link 00958 in 3D model. Treated with IL-17A resulted in increased epidermal thickness and higher numbers of the Ki67 positive cells in the 3D model. Both single molecular in situ hybridization and RT-qPCR validate that IL-17A significantly induced link 00958 in 3D epidermal equivalents. To explore the mechanism of IL-17A mediated regulation of the link 00958, we integrated the published available data from the ENCODE and identify the transcription that is binding with the promoter region of the link 00958. One of the transcription factors reviewed by this analysis is CBP beta, which is known to be activated by IL-17A signaling pathway. The knockdown of CBP by small interfering RNA resulting in a sufficient suppression at the protein level also lead to the impaired ability of the IL-17A to induce link 00958. Because P38 is a downstream effect of the IL-17A, our work shows there's a decrease of the total and the phosphorylated CBP upon the P38 expression inhibition. Moreover, IL-17A induced link 00958 was significantly suppressed by P38 inhibition, suggesting that IL-17A regulates CBP pathway through P38 pathway, thereby moderate the link 00958 expression in the keratinocytes. To determine the effects of link 00958 on the gene expression landscape in the keratinocytes, we inhibit the expression and use a series of pore of sign RNA. And in this heat map, they show boundaries of the genes, which is downregulated after knockdown of the Link0958. After that, we perform gene ontology and GSEA analysis, which revealed that significant enrichment of the genes related to cell proliferation. Furthermore, we analyzed the effects of the Link0958 depletion on keratinocytes proliferation. We perform immunofluorescence analysis to detect the decreased expression of Ki67 in Link0958 depleted keratinocytes. In line with this, a real-time analysis of cell conference using the IncuCyte is showing that there is a significant decrease of the cell growth in the Link0958 depleted keratinocytes. Moreover, we perform EDO assay analysis, demonstrate that depletion of the Link0958 lead to a reduction of a proportion of the cells entering to the S phase. Interestingly, treatments with IL-17A result in the accumulation of the cells in S phase and G2 phase, while the proliferative effects of IL-17A was counteracted by inhibiting of Link0958. To get insight to which signaling pathways through the Link0958 regulate keratinocytes proliferation, we determined there's a relative site-specific phosphorylation of 37 human protein kinases in Link0958 depleted or controlled keratinocytes. Interestingly, the analysis identified P38 as one of the top kinases with a decreased phosphorylation in Link0958 depleted keratinocytes. To test if Link0958 induced P38 phosphorylation could mediate IL-17A induced keratinocytes proliferation, we first validated by West blot and show the inhibition of Link0958 suppressed both baseline and IL-17A induced phosphorylation of P38. We perform EDO assay again in the keratinocytes and confirm previously reported the role of IL-17A as an inducer of keratinocytes proliferation. Co-treatment IL-17A with P38 inhibitor lead to a reduction of the proportion of the cells entering into the S and G2 phase, indicating that P38 inhibition abolished IL-17A induced proliferation in keratinocytes. Now, I would like to conclude this study. Our work identified IL-17A getting involved. Our work identified Link0958 as getting involved in a positive feedback circles in the psoriasis, and IL-17A induced overexpression of the Link0958 through the P38 and the CBP pathway that can lead to further activation of P38 and increased proliferation in the keratinocytes. In the end, I would like to thank my supervisors and all our group members' contribution, and thanks all the grant sources to support this study. Thanks for your attention, and I'm open to any questions.
Yeah, thank you very much, Longlong. Also, this was a really impressive presentation. Now we come to the Q&A session, and if you allow me, Longlong, I would just follow up a bit on why I said that I feel that there might be a link between the two manuscripts. What would be your proposed function of this Link0958 in the context of wound healing? Would you see any relevance there?
I would say it's a really interesting question. For our study, we haven't investigated for wound healing so much. And as a psoriasis disease, which is mainly regulate inflammation a lot instead of like skin regeneration. So I don't have an idea about, but it could be interesting to make any collaboration with this wound healing project.
Elodie, what do you think the other way around? Could your metabolic finding, let's say, also have an impact on inflammation? What's the link here? You also showed this IL-1 beta positive.
I have not seen any data neither on long non-coding RNA and wound healing, but I think it's totally possible. I think just we did look. Yeah, and for sure, the metabolism is very important for keratinocyte and fibroblast during wound healing regeneration. So yeah, for sure, I think it would be very nice to take a look.
Thank you. There are also questions in the chat, and one first for Elodie. So it seems that in your histological staining in the back skin, you have hair follicles which contain stem cells, which could contribute to wound healing. And then, of course, you do not have this hair follicle into the velvet. So how do you think this can interfere with the wound healing?
So in fact, we have this hair follicle in back and in back skin and in the velvet skin, but there are some differences between these two hair follicles. So the hair follicle in the velvet are more closer and smaller than the hair follicle that we can find in the back skin. So they are here. And for sure, the hair follicle have stem cell, epithelial stem cell, and mesenchymal stem cell. We call them the HFDSC. So this is one of my previous paper where we found that the HFDSC, so the fibroblasts in the bottom of the dermal papilla in the hair follicle that are responsible for the regeneration of hair follicle at each hair cycle is not responsible for or not fully responsible for the regeneration of the hair follicle after a wounding. So we know that it's all the fibroblasts into the dermis and not only the fibroblasts, more stem cell inside the hair follicle. I don't know if I was clear.
Yeah, it was clear. There is another question for you, Elodie, about the Schwann cells. It seems that there is a difference between back and velvet skin. Any idea why?
Yeah, it's true that it's one of the main difference as well. So we didn't look at too much just because, so there is a difference when we do the Pearson correlation, but there is maybe a difference because there is a difference in terms of number. So I think we could find way more or greater number of Schwann cell in the velvet in comparison with the back skin. And we think that is why the Pearson correlation is different. Because when we check the differentially expressed gene in velvet and in back, only in Schwann cell, we didn't see too much difference. So we think more it's a question of number of cells that we captured for the single cell RNA-seq, more than a true functional difference in these two tissues.
Thank you very much. Any other question?
I would have one final question for Longlong, maybe. Do you know anything about the kinetics? I-17A induces this link. Would you think that's a factor that contributes to early psoriasis, or is it rather a factor for continuous and chronic inflammation? Where do you see it? How stable is this induction? How quickly is it gone? Do you have any idea here?
Do you mean the mechanism, for example, P30 pathway, is regulate the status of
the upregulation of the link itself? I mean, okay. Right? It's IL-17 that upregulates it, but do you know how stable that upregulation is? Is it like a peak coming and going?
Yeah, I think this is a very interesting question, and it's also need to a lot of investigation for the human biopsies. And actually our biopsies, which is taken from the mixed ages and the mixed sex from both different, there's a quite wide range. So basically we will see the upregulation in psoriasis, skin, which is quite abundant evidence. We didn't see the very age-dependent expression for this. So we'll see maybe like this link, which is really getting involved in the pathogenesis, not only depends on the age or something like that.
Good, thank you very much, you two. So we know we will hear a lot more from both of you. Elodie has quite some other cell types to study, and Longlong quite some other non-coding RNAs. So we are sure we will hear more about you. Thank you very much for your participation today. With this, we would just like to mention the next one, which will be really a highlight, I think, at 7th of June. Ellen versus Emma in the inside versus outside in theory, which is long ago, since the first times of genetics, and you might guess what kind of disease they will be talking about. Then we will also shortly announce the ESDR, which will be next year because of the ISID, next year in September in Lisbon. It is still a way to go, but we are heavily working on that, on the preparation of having a great meeting here. So please already mark that into your calendars. With this, all of you, thank you very much for listening to today's Kitchen, and we look forward to seeing you soon again at another episode. Bye-bye.